All right, guys, welcome back to Underground Science. In this video, we'll be talking about protoporphyrin synthesis, okay? Because we need to know this when we talk about sideroblastic anemia, all right, in our next video. So let's talk about how protoporphyrin is synthesized. Okay, so let's focus on the mitochondria first. So what happens is, and remember, this is happening and you're developing red blood cells, okay? All right, so all this is happening inside your erythroblasts. Okay, so first we have our mitochondria, right? So what happens in the first step of protoporphyrin synthesis is actually occurring in your mitochondria. You get succinyl-CoA and glycine, right? Together form amino-levulinic acid, or ALA. And they form this or this is formed, right? Amino-levulinic acid is formed by the enzyme amino-levulinic acid synthase, right? ALA synthase, or ALAS, right? For short. Now, it's really important to know that you need vitamin B6, okay? This enzyme needs vitamin B6 to function. So, of course, if you have a vitamin B6 deficiency, this enzyme won't be able to function and you won't be able to do protoporphyrin synthesis. Okay, so that's really important to know. Now, in this video, we're going to add in many diseases, okay? In terms of diseases affected by what defect in each enzyme. Okay, so if ALAS is defective, right? If aminolevulinic acid synthase does not work or is defective, you get X-linked sideroblastic anemia. Okay, sideroblastic anemia. All right, and we'll talk about sideroblastic anemia in our next video. So that's amino-levulinic acid synthase. It needs B6 to function. It takes succinyl-CoA and glycine and makes amino-levulinic acid. If amino-levulinic acid synthase is dysfunctional, you get X-linked sideroblastic anemia. All right? Cool. So now amino-levulinic acid moves out of the mitochondria into the cytoplasm. Okay? From there, ALA, right, is converted into porphobilinogen via ALA dehydratase. Okay? So via amino-levulinic acid dehydratase. Again, it's important to know that this enzyme, amino-levulinic acid dehydratase, right, needs zinc in order to function. So ALAD, right, short for amino-levulinic acid dehydratase, needs zinc to function, okay, and it converts ALA to porphobilinogen. Now, what if your ALAD enzyme is defective? Then what can you get? So if your amino-levulinic acid dehydratase enzyme is defective, then it's actually not about what you can get, but how does it even get defective? It gets defective via lead poisoning. Okay, so if you have lead poisoning, this can lead to a dysfunctional amino-levulinic acid dehydratase enzyme, which means you won't be able to do porphobilinogen synthesis. Or sorry, protoporphyrin synthesis. My apologies. All right. So now we have porphobilinogen. Porphobilinogen is converted to hydroxymethybilan, okay, via the enzyme porphobilinogen deaminase, okay, which is right here. Now, if this enzyme is defective, what do we get? We get acute intermittent porphyria, all right? So if this enzyme is defective, we get acute intermittent porphyria, all right? So that's important to know too. So now we have synthesized hydroxymethybilan. Now hydroxymethybilan is converted into uroporphyrinogen 3, okay, via uroporphyrinogen 3 synthase. Okay, so that enzyme is not hard to remember. Now, if this enzyme is defective, right, what do we get? Okay, so if uroporphyrinogen 3 synthase is defective, then we get congenital, right, erythropoietic porphyria. Okay, so if uroporphyrinogen 3 synthase is defective, we get congenital erythropoietic porphyria. All right, that's important to know also. Now, once uroporphyrinogen 3 converts hydroxymethybilan to uroporphyrinogen 3, Uroporphyrinogen 3 gets converted into coproporphyrinogen 3 via uroporphyrinogen 3 decarboxylase. So again, if this enzyme, uroporphyrinogen decarboxylase, right? If this enzyme is defective, what do we get? We get porphyria, cutánea, tarda. Okay, so that's important to know also. So now we have coproporphyrinogen 3. This moves into the mitochondria again. Okay, so then now coproporphyrinogen 3 gets converted into protoporphyrinogen 9 via coproporphyrinogen oxidase. Okay, coproporphyrinogen oxidase. Okay, so now, so again, coproporphyrinogen oxidase converts coproporphyrinogen 3 into protoporphyrinogen 9. If coproporphyrinogen oxidase is defective, then we get coproporphyria, okay? Or also specifically hereditary coproporphyria, all right? So that's when coproporphyrinogen 3 is, or coproporphyrinogen oxidase is defective, all right? Then you get hereditary coproporphyria. Now, protoporphyrinogen 9, right, gets converted into protoporphyrin 9 via protoporphyrinogen oxidase. If this enzyme is defective, then you get variegate porphyria. All right, then you get variegate porphyria. Okay, so that's important to keep in mind also. So now that you have protoporphyrin 9, right, the last step, you need iron. Okay, you need iron in order to make heme, right? So what happens is the iron combines with protoporphyrin 9 to make heme via the enzyme ferrochelatase. All right. So what happens if what can basically inhibit ferrochelatase? Lead poisoning. Okay. So remember, lead poisoning not only can inhibit ferrochelatase, it can also inhibit amino levulinic acid dehydratase, right? Because remember, you get glycine and succinyl CoA that combines to make amino levulinic acid, right? Via amino levulinic acid synthase. Then ALA, right, gets converted, as we know, into porphobilinogen, right? So remember, we get ALA converted into porphobilinogen via the enzyme ALA dehydratase, right? And this can also be inhibited by lead poisoning. Okay, so that's important to remember too. So that was the whole goal, right? Is to synthesize heme. Okay, you need to synthesize protoporphyrin first, though, so that iron can move in to the mitochondria, right, and combine with protoporphyrin 9 and form heme. And then this heme, right, can get incorporated into hemoglobin, right? If you combine globin with this, and then you get your hemoglobin, right? So you can imagine if you don't have enough iron, then you can't make heme, right? Which means you can't make enough hemoglobin. Therefore, you would have microcytic anemia. Okay, so that's why iron deficiency anemia is a type of microcytic anemia, and sideroblastic anemia is a type of microcytic anemia, because in sideroblastic anemia, right? Remember, what was X-linked sideroblastic anemia? What enzyme was that related to? ALAS, right? ALA synthase. So if you have defective ALAS enzyme, then you can't properly do the protoporphyrin synthesis pathway. So you can't make protoporphyrin, which means you can't, which means, right? So what will that lead to, right? We'll talk about this when we talk about sideroblastic anemia. But let's say you can't make protoporphyrin, okay? So iron will keep on coming into the mitochondria, right? But you don't have protoporphyrin for iron to combine with to make heme. So that's the issue here, right? So that's still going to lead to decreased heme production because you can't produce protoporphyrin, which is a component of heme, leading to microcytic anemia. All right. So that was protoporphyrin synthesis. Okay. Hopefully this video made sense. Next, we'll talk about sideroblastic anemia. All right. So see you guys there. Thank you guys for watching.